and I am a senior here at UK, majoring in biology and minoring in pharmacology. I do reproductive endocrinology and toxicology research under Dr. Patrick Hannon in the OBGYN department at the College of Medicine, and our research focuses on environmental toxicants and their effects on female reproductive health. Emerging science suggests that unavoidable exposure to different chemicals may cause infertility. Some of these toxicants include a family of chemicals called phthalates. Phthalates are used as solvents and plasticizers in a multitude of common consumer products, including personal care items, cosmetics, food and beverage containers, car upholstery, and even in the building materials found in our homes, workspaces, and large cinemas like this. And because of their widespread use, avoiding exposure is nearly impossible. Humans are also exposed to multiple different types of phthalates on a daily basis, so this study utilizes an environmentally relevant phthalate mixture, which is a unique approach because the majority of other studies investigate only single phthalate exposures. And you can see the composition of this mixture in the gray table on the left of the slide. So why does this matter? This matters because previous research suggests that phthalates are able to directly target the ovary. And because phthalates have been known to disrupt endocrine and reproductive systems, exposure may be having negative effects on fertility and overall reproductive health. Studies also suggest that women who are actively seeking treatment at IVF clinics due to fertility issues experience a lower quality of life. And because ovulatory defects are the leading cause of infertility in women, meaning the egg is not properly released from the ovary, we investigate the effects that these chemicals have on the ovulatory process directly. So collectively, having a better understanding of how phthalates potentially hinder ovulation could be beneficial to the overall health care of women. So briefly, ovulation occurs when the egg is released from the pre-ovulatory follicle, which are the functional units of the ovary that house the egg. As you can see in the graphic in the bottom left corner, this process is initiated by the mid-cycle luteinizing hormone surge, which I will refer to as LH, or clinical treatment of human chorionic gonadotropin, which I will refer to as HCG, and is essentially just a more potent analog of LH. This surge of LH or HCG also induces prostaglandin production. Prostaglandins are integral molecules that drive ovulation by causing inflammatory-like processes. These LH or HCG-induced increases in prostaglandins are required for successful ovulation, and inhibiting prostaglandins results in failure of ovulation. Even taking a common NSAID like aspirin or ibuprofen, which inhibits prostaglandin production, at the appropriate timing can inhibit ovulation in women. So specifically, this study investigates the effects of this environmentally relevant phthalate mixture on prostaglandin production. So we essentially did this study by isolating individual ovarian follicles and putting them into culture with different treatment groups. Those treatment groups included DMSO alone, which is not an ovulatory stimulus, HCG alone, which is an ovulatory stimulus and is that more potent analog of LH that I previously mentioned, and HCG plus increasing doses of the phthalate mixture. We then visually assessed ovulation and measured prostaglandin levels. So in the top right corner, we have our ovulation assay, and it is our most striking data. The y-axis represents ovulation percentage, and the x-axis represents our different treatment groups. DMSO is always represented by a white bar. The HCG ovulatory control group is always represented by a black bar, and the color bars represent HCG plus increasing doses of the phthalate mixture ranging from lowest to highest dose. As you can see, when the follicles were treated with no ovulatory stimulus, ovulation occurred at an expected 0%. When treated with the ovulatory stimulus alone, ovulation occurred at an anticipated 80%. But as we increase the dose of the phthalate mixture, we see a greater decline in ovulation rates, with the highest dose of the phthalate mixture being statistically equivalent to the group that did not receive the ovulatory stimulus and did not ovulate. So below the ovulation assay, we have graphs that represent the levels of two different prostaglandins, PGE2 and PGF2 alpha. The y-axis now represents percent change of prostaglandin levels relative to HCG alone, and the x-axis represents those same treatment groups. So here, when treated with the ovulatory stimulus alone, we see expected increases in prostaglandin levels. However, similar to the ovulation assay above, when we treat with the phthalate mixture, we see decreases in prostaglandin levels at essentially every dose of the mixture. So collectively, these data suggest that phthalate exposure may be contributing to ovulatory defects by decreasing prostaglandin levels. Again, this is concerning because ovulatory defects are the leading cause of infertility in women, and if we can better understand the mechanisms by which phthalates potentially hinder ovulation, we can provide better health care to women suffering from different reproductive disorders. I want to thank you all for listening, and I am happy to answer any questions.
Thank you very much. Excellent, Katie. I guess there are a lot of questions and again a lot of ignorance on my part, but the main question I've got is give me a sense of scale. So we've got 10 micrograms, 100 micrograms, and 500 micrograms per milliliter. But what does that look like? What does that relate to in terms of the hand lotion or whatever those things might be? How much engagement does a woman need to have with these in the wild, so to speak, in order to see these sorts of effects?
Right. So people have different exposure profiles based on numerous factors, including like lifestyle choices, different occupations, things like that. So exposure to different of these individual phthalates ranges anywhere from 0.12 to 30 microgram per kilogram per mil. In relation to our specific concentrations that we chose, we want to see a dose response curve here. So also, if you look at the concentration of the individual phthalates relative to its percentage of the mixture as a whole, the lowest dose of the phthalate mixture, which is 1 microgram per mil, is still below the highest level of that same phthalate found in follicular fluid from women. So this mixture is environmentally relevant in that sense. Does that answer your question?
Sure, you answered it in there, and I just lost it. So just bear with me for a second. So you just said the 1 microgram is that? I thought I heard you say that your average woman, it would be 0.5 or something to 30 micrograms.
Right, but that's for the individual phthalates, and this is for its percentage as the mixture. Yes.
Oh, I see.
Okay.
So which of these is sort of where an average American woman would be expected to have exposure of this combined nature?
So because of the statistics I just told you of like the average exposure, I would say the 1 and the 10 microgram. However, because people do have different exposure profiles, that can like vary.
Right, of course, but okay. So we're looking at the pink bar as sort of where,
yes.
Between yellow and pink is sort of where most folks are, but it's still having a significant impact already
exactly.
at those
yes.
levels. And so I know we're taking this beyond that, but I want to ask, conclusively demonstrating this, what would you see as the next stage? Is it creating a list of things to avoid for a woman who wants to conceive, or what would your recommended practical real world steps be for this?
That's a great question. So this study wasn't necessarily designed to make regulatory statements such as we should ban phthalates. However, certain countries, including like the US, Thailand, and countries in the EU, are starting to limit or ban certain concentration of phthalates in specific items, namely like children's toys. I just think it's very important for people to be aware of the chemicals that they're exposed to on a daily basis. I know personally, since getting involved in this research, I definitely start to look at the chemicals that are in the products that I'm using, like hairsprays and lotions, shampoos, perfumes, things like that.
Great. Thanks. Well done, Katie.
Thank you. Yes, if I could follow up on this. Is phthalate exposure the kind of thing that can be found, say, in our water supply and water supply to start with?
Yes.
Or is it only something that can be measured in human fluids or human cells or
yes.
animal cells?
So I actually have read some papers. Phthalates are able to bind and be in water. Actually, the most common route of exposure to phthalates is oral ingestion through like food and beverage containers.
So if it's in your plastic Tupperware, then it can be in the food that you're eating out of the plastic Tupperware, and therefore will end up in your water supply as a waste product, possibly. And you looked at mice.
Yes.
Are there other animals that you would use to do this kind of experiment on?
In our lab specifically, we do mouse work as well as human work. Due to my limitations of being in the lab as a student, I'm definitely more involved in the mouse study, but we are doing human work. And those are like follicular aspirates from humans, from women.
Yeah, a couple of questions. One, mainly related to your use of mouse.
Mm-hmm.
So, and this kind of follows up on a previous question about these concentrations of 1, 10, 100, 500 micrograms. Are those are relative to the mouse or relative to a human? So in other words, you know, a mouse is obviously not a human, much smaller.
Mm-hmm.
Are these concentrations expected to affect humans, or are they just demonstrated via the mouse?
Right, so this mixture was actually derived from urinary phthalate levels found in pregnant women enrolled in a study by our colleagues at the University of Illinois at Urbana-Champaign. So these concentrations are based off of human exposure. However, we do know the mouse is a good model for this, because if you look at our data, the HCG, which is the ovulatory stimulus, causes abundant increases in ovulation rates as well as prostaglandin levels.
And so, and that brings me to another question of curiosity. These treatments are based on the human gonadotropin, not the mouse. So you're telling me the human version of this substance affects ovulation in mice?
Yes, so humans and mice actually have really similar reproductive systems. So while this mixture was derived from levels from humans, we know that. Are you asking, like, are these exposures relevant?
No, what I'm asking is, are you using the human version of this hormone on the mouse, or are you using the mouse version of the hormone on the mouse?
Oh, I'm sorry, I see.
In addition to the chemicals.
Right. So the HCG, human. Okay.
Right.
That is a hormone that is. It acts on both the mouse and the human. It's the same hormone.
Thank you.
Sorry.